Hello, everyone. This will be our new lecture on determination of sex. Now, here are the objectives. At the end of the session, we would be able to cover and describe the human fetal development using the three-part alignment, and describe normal male and female development of sex, distinguish the genes involved in normal development of sex, discuss and describe the role of Y chromosome in development of sex. I've divided this topic into two parts. First one, I will describe all these objectives, and the second part will be more of complementary to this part one, where I will explain the molecular aspect of the central dogma for some of the essential genes involved in the sexual development. Now, a basic introduction to the fetal development. Now, one of the parts that are very important in this slide is the three-part alignment. The three-part alignment basically, what's the sex chromosomes that a person have, which is actually dependent on the fertilization process. And upon acquiring the sex chromosomes, then the development or the formation of the gonads, or what we call the internal genitalia. Now, once the internal genitalia is formed, then that will dictate the formation of the external genitalia. So the first part of alignment is the sex chromosomes. The second part is the gonads or the internal genitalia. And the third part is the external genitalia. For a presumptive male, this is how it should look like: XY followed by testis, and finally the external genitalia of a male. Whereas in a female, the sex chromosome would be XX, ovaries as the gonads, and the external genitalia of a female person. So what makes us male or female? Okay, here are some options: the presence of X chromosome, the presence of Y chromosome, the autosomal chromosomes, both autosomal and sex chromosomes. These are the options. I would like you to ponder these while we are going through the entire session. Prenatal development, and the first step in sex determination occur at the time of, try to answer this question, or as the slides goes on, you will probably be able to actually answer this question. Chromosomal sex makes things possible for future change. That is a fact. But during this time, the two sexes are indistinguishable. Okay, this is very important. We will look at what it means by indistinguishable. By the 6th week of human development, both 46XX and 46XY embryos have the same sex organs. The two sexes have a unisex gonads called undifferentiated gonads. Okay, this is what it means by indistinguishable. Where do undifferentiated gonads develop from? As time to time, this particular session, there'll be a lot of questions in a form of something like this. You will need to find the answers to these questions. Now, what are the main genes that are involved in the development? Is SF1. Okay, it's called stereogenic factor 1. Okay, it's a nuclear receptor gene, and the function of it is to activate hormone production, development of mature gonads, and adrenal glands. Now, I will also be highlighting in this format Okay, I will take some time to explain this first, and the subsequent one I will not be explaining, as you will have to decipher on your own. Okay, this is SF1 gene, locus 9q33.3, human SF1 spans 26.3 kb, 7 exons, including non-coding first exon, and it encodes 461 amino acids. So what does this mean? Now, SF1 gene is located on the genome on chromosome 9 long arm, position 33.3, and the size of the gene is 26.3 kb. It has 7 exons. The 7 exons includes a first exon which does not participate in the translation process. That's why it's a non-coding first exon. Now, the resulting amino acid that is formed from this gene is 461 amino acids in size. Okay, that is the meaning of this molecular biology of the central dogma for SF1. SF1 protein is a sphingolipid binding protein. Next gene that is involved in prenatal development is WT1, Wilms tumor protein 1. It is an active in development of both gonads and kidneys. Okay, and the molecular biology for WT1 is provided in this. It's located in 11p13, which says that similar to SF1, it is an autosomal gene. Okay, its size is 345 kilobase, 10 exons, and it encodes 429 amino acid. Now it is a zinc finger binding protein. Now whenever you have this motif, that means it can interact with DNA. The protein that is formed from this gene can interact with other DNA regions. Two sets of primitive ducts are available, which are called Wolffian ducts and Mullerian ducts. Wolffian ducts is the male duct system, whereas the Mullerian ducts is the female duct system. This duct system will eventually form the rest of the internal sex organs. What about the external organs? Both 46XX, 46XY embryos develop knob-like genital tubercle and urogenital groove, surrounded by specialized folds and swellings of tissues. As you can see, in early development of internal and external, which is the gonads and the external genitalia, any embryo is well equipped to become which sex? Is it a male or a female? Okay, you will need to answer this question on your own. Okay, this is at the 6th week, whereas this is at birth. Okay, as you can see, the blue indicates the Wolffian ducts and the red indicates the Mullerian duct. And here you have the mature male external and internal genitalia. Here is the female external and internal genitalia. How then we differ? Male hormones need to intervene at later stages of the fetal life. If no intervention, what happens? There will be a default kind of setting which will kick into action. Okay, in a presumptive male, okay, the second step happens. Okay, the first step that now when during the formation of the first part of the alignment, which is the sperm and the ovum. During fertilization, that is the first step that dictates whether which chromosomes do you get. Now the second step happens at the 7th week of development. Now the gene that is involved here now is SRY, sex determining region Y. And this gene's product makes things happen. Okay? The SRY gene molecular biology is given in this. It is a very unique gene as it is a intronless. Okay? It does not contain any introns. It's 204 amino acid in size. It's a very small gene, 3.8 kilobase, and it's located under Y chromosome at the short arm of the Y chromosome. SRY gene is also known as the testis determining factor, TDF. It's a DNA binding protein and it is transactivated by WT1. It is also part of a DNA binding protein kind of proteins and is a member of the high mobility group. Okay, as you can see, the high mobility group. Okay, it also triggers the inner parts. Okay, the formation of the inner parts of the gonadal ridges to begin developing into testes. So basically, SRY gene is very much involved in the formation of the testes, which is the gonads for the male. SRY gene is necessary but not sufficient for the differentiation of mammalian male gonad. Autosomal and X-linked genes are very much involved in how this differentiation takes place. SOX9, or what is called as SRY box 9 gene. Okay, here is the molecular biology for this. Now, for no reason, please do not memorize this molecular biology of all these genes. These are only to guide you in understanding that these are all autosomal genes, and when it's given X or Y, then it's become a sex linked genes. Okay, and the size of the gene followed by the size of the protein. Now, SOX gene shares a special DNA binding sequence with SRY. Remember, SRY is a high mobility group kind of protein. Now, the binding of SOX9 to DNA bends the DNA at an angle of 70 to 80%. When this type of bending happens, what do you think will happen at the molecular level? We have covered the first step and the second step. Okay, now it's the third step in male development. First step was during fertilization. Second step during the 7th week of development. Now the third step occurs when the somatic cell in the fetal testis begin to produce two different hormones. What are the hormones? They are called anti-Mullerian hormone. Anti, from the word anti itself, you know it's against the female type of hormone, Mullerian hormone. So anti-Mullerian hormone, AMH, and the other type of produced male sex hormone, testosterone. Obviously, the male have to have the testosterone hormone in order to make things possible. What cells produce which hormone and their roles? Okay, this is your homework to find out which cells produces anti-Mullerian hormone and which cell produces testosterone. The AMH, anti-Mullerian hormone, found in the autosomal chromosome, it is chromosome 19, long arm. Gene functions only in the presence of Y chromosome. Okay, the gene is only activated in the presence of a Y chromosome. AMH activity is regulated by SF1. The regression of the Mullerian ducts requires a normal AMH receptor. Every hormone interacts with a type of a receptor. If you have the receptor, then only the hormone will actually work. If you do not have a receptor, or the receptor is mutated, some kind of mutation happens in the DNA sequence, then the actual receptor is mutated, then no matter how much of hormones you have, it will not interact correctly with the receptor. Now, the receptor is also encoded by a autosomal gene, which is located on chromosome 12 long arm. Now, if you do not have an proper interaction, you will probably have a syndrome called persistent Mullerian duct. Now, if the appropriate tissues are responsive to the hormones and the following changes occurs by the 7th week, now internally the Mullerian ducts begin to degenerate, because in a male, you will probably need to get rid of the female ducts. Okay, under local influence of the testosterone, remember the testosterone is being produced, the Wolffian duct begins to form prostate glands, seminal vesicles, and vasa differentia. Neural pathways in the hypothalamus controlling menstrual cycle is altered. If the appropriate tissue again is responsive, what will happen in the 7th week is a very potent derivative of testosterone. Okay, it's called dihydrotestosterone, organizes the shapes of the external genitalia. Third part of alignment, no? When the gonads are all in a proper order, when the hormones are being expressed, therefore you will have the third part of alignment properly occurring. If this particular derivative is not formed properly, then you will have problem with the external genitalia. Testosterone is converted to dihydrotestosterone by two variants of the enzyme 5-alpha reductase. Now, in human, the mutation in this particular gene contributes to a lot of sex ambiguity in male. The genital tubercle and the surrounding folds lengthen and fuse to form penis. This is the external genitalia. Swelling that are nearby will develop and fuse to form the scrotum. So these are the external genitalia formed by the help of the 5-alpha reductase. Okay, because the 5-alpha reductase ensures the formation of dihydrotestosterone. What makes the female external genitalia? In a presumptive female, the second step happens at the 12th week of development. The development of ovaries from the outer part of the genital ridge is caused by a female hormone. You would probably guess, okay? Now, DAX1, dosage-sensitive sex reversal adrenal hypoplasia is a key player in this development. Okay. DAX1 encodes a nuclear hormone receptor that is related to SF1. DAX1 functions as an anti-testis gene by opposing the effects of SRY. DAX1's molecular biology is given here. DAX1 is a sex-linked. Okay, it's found on the X chromosome, short arm of the X chromosome. It has got 2 exons and encodes 470 amino acid size protein. It's not a big gene, but it's only 1.59 kb. There are some X-linked loci and autosomal loci that are required for normal female sexual development. We know for a fact that 2 X chromosomes are required for ovarian maintenance. So if everything goes correctly and all the internal organs are responsive, then these are the changes that will happen in a female. The Wolffian tubes regresses in the absence of testosterone secretion. Why testosterone secretion is not there? Y chromosome is not there. The Mullerian tubes form the female ducts in the absence of anti-Mullerian hormone. Anti-Mullerian hormone also requires the Y chromosome, presence of the Y chromosome. So therefore, you will not have anti-Mullerian hormone. So therefore, the Mullerian duct will thrive. The forward parts become the oviducts and part of the vagina. The external genitalia develops clitoris from the genital tubercle, small and large labia from the surrounding folds and the swellings, lower part of the vagina from the urogenital growth. So the next question would be, when will the sex of the fetus will be apparent? Okay, at what week do you think the sex of the fetus will be apparent? Okay, this is just a guess. Okay, an educated guess. Okay, so you will probably have to identify the particular time when do you think the sex of the fetus will be apparent. Now, in a summary, this is what we have actually gone through. Okay, the genital ridge. Okay, it's being governed by these two basic or the primary type of genes that will ensure, okay, with the help of the next group of genes, okay, and the SRY in a male, if you have the male, if you have the Y chromosome, then the things will take on to become the development for male, okay. And then in the male, there are type of cells which actually contribute with the help of the, again, with the SF1 to form certain type of hormones. And these hormones will actually allow the regression or the progression of certain ducts to form the internal gonads and the external genitalia. Similarly, in the female, similar parts happen. So this diagram is a very useful diagram which summarizes whatever we have discussed. So with that, I conclude today's sharing session. Thank you.